Think about working out for a second. Like you go to the gym, you lift some weights, you put stress on your muscles, and they get stronger. It's just a foundational rule of how our bodies operate, right? Cause and effect. You break the tissue down slightly with mechanical stress.
And body just builds it back up with more capacity.
Yeah.
Yeah, it's basic biological adaptation. The system recognizes those microtears from the load, and it overcompensates during the repair phase.
Right. So you come back stronger the next time you encounter that weight, your muscles can actually handle it.
Exactly.
But I want you to imagine if that equation was entirely flipped. Like, what if the very mechanical act of moving, just walking across a room or, simply existing in gravity, was the exact thing destroying the muscle tissue?
It's a terrifying thought, honestly.
It really is. And welcome to this deep dive. Today, we are exploring a biological paradox that is, frankly, as fascinating as it is devastating, which is muscular dystrophy, or MD.
Yeah, and we have a really massive stack of historical medical records today, along with some incredibly complex genetic data and some very intense modern pharmacology to unpack.
We really do, because our mission today goes way beyond just, listing medical symptoms. We really want to unpack the pathophysiology of MD. We're going to explore how genetic defects literally break down human muscle at the microscopic level.
Right. And to do that, we'll trace the rather dramatic 19th century history of its discovery. And then we'll explore the, frankly, massive genetic mechanisms operating inside your cells right this second, and navigate the fierce modern controversies surrounding how we're actually trying to cure it.
Right, because understanding MD isn't just about understanding one isolated condition. The sources make it clear that MD has basically become the ultimate proving ground for the future of all genetic medicine.
It absolutely has. Everything we learn here, like all the regulatory battles and the genetic tools, it's all going to dictate how we treat a whole host of other diseases down the line.
Exactly. But to understand what MD is today, we kind of have to rewind first. We need to look at how the medical community originally defined it, which, according to our sources, involves a pretty contentious 19th century naming dispute.
Yeah, naming disputes in medicine are always intense. So the disease most people picture when they hear muscular dystrophy is Duchenne muscular dystrophy, or DMD.
Right.
And it bears the name of this French neurologist, Guillaume Benjamin Hamon, Duchenne de Boulogne. He published this series of incredibly detailed seminal case studies back in 1868.
And he was focusing on what he called pseudo-hypertrophic muscular paralysis, right?
Yes, exactly. And that term pseudo-hypertrophic is really key here. It means false enlargement. So the calves of these patients, they actually look muscular and large, but they were terribly weak.
Right. They look strong, but it was basically an illusion. And Duchenne didn't just, observe these patients from across the room. He actually invented an entirely new medical device just to study them.
He did. He developed this specialized needle biopsy device he called the Import piece, which it essentially translates to a hole punch or a cookie cutter.
Wait, like a literal cookie cutter for living muscle tissue?
Basically, yeah. I mean, before this, if a doctor wanted to examine muscle tissue microscopically, they generally just had to wait until an autopsy.
Oh, wow.
Yeah, so Duchenne's tool was this massive leap forward. He could actually sample living muscle tissue repeatedly from the exact same patients over time.
So he's tracking it in real time.
Right. He could document the progression of the disease histologically under a microscope while the patient was still alive. And, combine that with his pioneering use of clinical photography, recording his patients' declining postures, and his sheer thoroughness just locked his name into the medical literature forever.
Well, let me play devil's advocate here based on the historical records, because the history of medicine can be pretty unfair. Duchenne got the name, but he was absolutely not the first physician to discover or even describe this disease.
Oh, far from it, yeah. 17 years before Duchenne's major publication. So in 1851, an English physician named Edward Marion described the exact same disease.
17 years earlier.
Yeah. He presented this highly meticulous paper to the Royal Medical and Chirurgical Society of London. Marion also looked at muscle tissue under a microscope, and he made this brilliant, entirely non-obvious deduction for the mid-19th century.
Which was what?
He noticed that the sarcolemma, which is the protective outer membrane of the muscle cell itself, was breaking down.
And that observation is the linchpin, right? Because by seeing the actual membrane destroyed, Marion proved the destruction was originating in the muscle fiber itself.
Exactly. He proved it was a primary muscle disease. And we have to view this through the lens of the 1850s, when the dominant assumption was that all paralytic diseases like polio were neurological.
Right. The medical establishment just assumed the nerves were failing to send the signal, causing the muscle to waste away.
Right. But Marianne said, no, the nerves are completely fine. The muscle is literally destroying itself. But, despite getting the science right first, history credited Duchenne.
It seems like eponyms just reward publicity and comprehensive documentation over actual priority. I mean, Marion gets the microscopic mechanism right, but Duchenne brings the photographs and the fancy biopsy punch, and he wins the naming rights.
Pretty much.
But speaking of physical signs, we really should bring Sir William Gowers into the picture, too. In 1879, he described what we now call Gowers' sign.
Yeah, and that remains one of the most reliable, though frankly heartbreaking bedside diagnostic clues in medicine today.
How does it work?
Well, when children with this disease are placed on the floor and asked to stand up, their hip and thigh muscles are simply too weak to push them straight up. So instead, they roll over, push their hips into the air like a downward dog yoga pose, and then they literally walk their hands up their own shins and thighs.
They have to use their arms to climb up their own bodies just to reach a standing position.
Exactly. And over 140 years later, pediatricians still look for that exact physical maneuver in clinics. It paints a remarkably consistent clinical picture over time.
Yeah.
And really, before the 1980s, muscular dystrophy was defined entirely by these clinical exams and what you saw in a biopsy. It was a pathological signature.
Meaning doctors were just looking for a specific pattern of damage.
Right. Progressive weakness, perfectly normal nerve conduction, and this repeating microscopic cycle. Muscle fiber death, a failed attempt at regeneration, and eventually the replacement of healthy muscle tissue by fibrous connective tissue and fat.
And that fat replacement, that's the pseudohypertrophy Duchenne noticed.
Yes, exactly. The calves look massive, but it's not muscle. It's essentially scar tissue and lipid cells just filling in the empty space where the muscle died.
Wow. But then molecular genetics arrives, right?
Yeah.
And it just blows that unified, neat little 19th century picture wide open. We suddenly realize MD is not one disease.
Not at all.
It is this massive umbrella term for more than 30 genetically distinct disorders. And the real shocker here is that they have completely different root causes. Like, some are caused by missing structural membrane proteins, others are caused by missing enzymes.
Yeah, and myotonic dystrophy, which is another major form, is actually caused by toxic RNA accumulating inside the cell nucleus.
Right, and that toxic RNA acts like a sponge, trapping vital proteins so the cell can't function. It has absolutely nothing to do with a broken cell membrane.
Right. Completely different mechanism.
So I actually have to push back on the terminology here based on all this. If MD encompasses 30 different genetic diseases with entirely distinct root causes, is muscular dystrophy even a useful medical term anymore? Or is it just a historical bucket we lazily group things into because they happen to cause weakness and look similar under a microscope?
That is a hotly contested space among geneticists right now.
Yeah.
Is MD a shared mechanism or just a shared pattern? And the consensus is increasingly that it's just a pattern. It's a historical umbrella term. And this is vital for you, as a listener, to understand if you are ever looking at modern clinical trials or treatments. A gene therapy designed to fix a structural membrane protein does absolutely zero good for a patient whose disease is caused by toxic RNA. You have to know the specific broken mechanism.
Right. And finding that specific broken mechanism takes us straight into the 1980s, because for over a century, doctors were essentially just looking at the aftermath of a fire, right? The scar tissue, the fat, the weakness.
Yeah.
But to actually stop the fire, they needed to find the spark. And they couldn't do that until technology advanced enough to let scientists peer directly into human DNA.
Which leads to the massive breakthrough between 1986 and 1987. A laboratory led by Louis Kunkel at Boston Children's Hospital accomplished something monumental. They used a technique called positional cloning to find the gene responsible for Duchenne MD.
Positional cloning. It's one of those terms that sounds pretty dry, but the reality of it is actually mind-bending.
It really is.
Yeah. I mean, imagine trying to find a single typo in a massive library full of encyclopedias, but you don't have a search engine. You don't even know what the book is about. You just have this incredibly vague clue about which floor of the library the book might be on.
That's a great way to visualize it.
Yeah.
They had to painstakingly map the location on the chromosome based entirely on inheritance patterns before they knew anything about what the gene actually did.
Wow.
Yeah, they eventually narrowed it down to the short arm of the X chromosome, specifically a location known as XB21. And once they found the gene, they identified the protein it produces, which they named dystrophin.
Now, the sources highlight a core idea here that we really need to emphasize for the listener. This gene is enormous.
Enormous.
In fact, the dystrophin gene is the largest known gene in the entire human genome. It spans roughly 2.2 million base pairs of DNA and contains 79 different exons, or coding regions. It takes up almost 0.1% of your entire genome dedicated to just this one single protein.
And the sheer absurd size of this gene is actually the root of the problem. It presents this enormous physical target for spontaneous mutations. It's basically a game of microscopic probability playing out during cell division.
Wait, so you're saying it's not that the DNA sequence for dystrophin is unusually fragile or prone to breaking?
Not at all. It's just a gigantic target on a dartboard. When the body is copying DNA during sperm or egg formation, random copying errors naturally happen. The genetic machinery occasionally drops a letter or duplicates a sequence.
Right.
Because the dystrophin gene is so incredibly large, those random everyday errors are statistically far more likely to land inside it than inside an average-sized gene. This explains why roughly 1/3 of all Duchenne cases arise from brand new de novo mutations.
So they aren't inherited from a carrier mother at all?
Nope. They just happen spontaneously in that one specific embryo.
Okay, that covers the gene itself, but let's talk about the protein it makes, because here's where the mechanical reality of the disease becomes so clear. Think of the dystrophin protein as a molecular seatbelt, or like a high-tension shock absorber. It sits right inside the muscle cell membrane, and its job is to physically anchor the internal skeleton of the cell, the actin filaments that generate the pulling force, to a complex of proteins embedded in the membrane.
Right, and that embedded group is called the dystrophin glycoprotein complex, or DGC. And the DGC connects all the way out to the extracellular matrix, which is the connective tissue outside the cell.
So you basically have a physical tether spanning from the deepest inside part of the cell straight through the membrane to the outside environment.
Exactly.
Every single time you flex your bicep or, just take a step, this molecular seatbelt absorbs the mechanical stress. It distributes the force of the contraction, so the fragile cell membrane doesn't just tear apart under the strain.
And without dystrophin, that entire mechanical structure is compromised. When the muscle contracts normally, the sheer force of its own movement simply ruptures the unprotected membrane. Marion saw it under his 19th century microscope, and now we finally know why it happens.
The actual mechanism.
Right. And once the membrane tears, the outside environment rushes in. Specifically, calcium.
And calcium is normally kept at incredibly low concentrations inside a resting muscle cell, isn't it?
Extremely low. But when that membrane tears, a massive wave of calcium floods in. And that calcium influx acts like a biochemical alarm, triggering destructive enzyme cascades proteases that begin digesting the muscle fiber from the inside out.
So the cell essentially eats itself and dies.
Yes.
Yeah.
And this molecular mechanism explains one of the most tragic clinical realities for families facing this diagnosis. Children with Duchenne walk completely normally at first. They hit their early milestones perfectly.
Right, because the structural defect is entirely invisible until the child grows enough to start putting real significant mechanical load on their muscles by walking, running, and climbing stairs.
Exactly. The defect hides until the muscle is truly subjected to the forces of gravity and resistance. And even then, the body mounts a fierce defense. Muscle tissue has these specialized stem cells called satellite cells. Their entire job is to jump in and repair damaged fibers.
But they can't keep it up forever.
Right. For the first few years, they keep up with the damage. But over years and years of continuous tearing, calcium flooding, and repairing, that regenerative reserve simply gets exhausted. The satellite cells give out, and the muscle finally surrenders to scar tissue.
It's devastating. But if this giant molecular seatbelt is so prone to breaking due to copying errors in the DNA, we have to ask, does how the gene breaks dictate how bad the disease is?
It absolutely dictates the severity.
Yeah.
And it leads to one of the most elegant, if somewhat counterintuitive, rules in modern genetics. It's called the reading frame rule.
The reading frame rule.
Yeah, Alan Monaco and his colleagues proposed it back in 1988, and it remains a true cornerstone of how we understand this disease today.
Let's break this down for the listener, because the logic here is honestly brilliant. To build a protein, your cellular machinery reads the DNA code in groupings of 3 letters at a time. Those 3-letter groups are the reading frame. So, imagine a sentence made entirely of 3-letter words. The fat cat ate the rat. If a random mutation deletes just 1 or 2 letters, say it deletes the F and the A in FAT, everything shifts over to fill the gap. The sentence becomes, the TCK ate a tet her at total gibberish.
Absolute gibberish.
Okay.
And the cell's machinery can't read it, so it panics and aborts the process. It produces zero functional dystrophin.
Yeah.
And the result of that is the severe, rapid progression of Duchenne muscular dystrophy.
Okay, but, however, imagine a deletion happens that removes a much larger chunk of genetic material, but it happens to remove an entire three-letter word. Say the mutation deletes the entire word FAT.
Right.
The sentence now reads, "The cat ate the rat." It's shorter. It's missing original information. But the underlying reading frame remains perfectly intact. The sentence still makes logical sense to the cell. Hold on, I want to really emphasize this for anyone listening who might be confused because it completely goes against common sense. Are you saying a patient who is missing more actual genetic material, like a larger dilution in their DNA, can have a significantly milder disease than a patient missing just one or two tiny letters?
It defies basic intuition, I know, but yes. Because the larger deletion preserved the reading frame, the cell doesn't abort the process. It reads the code, and it produces a shortened, partially functional dystrophin protein.
So it's a seatbelt that's a bit too short, but it still buckles?
That is exactly it. And this results in what we call Becker muscular dystrophy. Patients with Becker have a much milder course. They can often remain ambulatory, walking well into their 40s or even 60s, whereas patients with Duchenne typically lose the ability to walk in early adolescence.
Wow.
Yeah. And the reading frame rule correctly predicts the disease severity in roughly 90% of all cases.
That's incredible. Now, before we move into how this exact rule sparked a revolution in modern drug development, we do need to clear up a major medical misconception found in the sources.
Oh, yes. This is important.
We know the dystrophin gene sits on the X chromosome, and because males typically have one X and one Y chromosome, a single mutated copy on their only X chromosome causes the full-blown disease, that is classic X-linked recessive inheritance.
Right.
But there is a persistent myth that female carriers who have two X chromosomes are entirely shielded from the disease.
Yeah. The traditional medical assumption was always that the second healthy X chromosome fully protects them. It acts as a perfect backup generator. But, cellular biology is rarely that neat and tidy.
Right. There's always a catch.
Early in embryonic development, every female cell randomly silences one of its X chromosomes in a process called lionization, or X inactivation.
And the cell shuts one off to avoid getting a toxic double dose of X chromosome genes.
Precisely. It basically crumples one X chromosome up into a tight ball so it can't be read. Usually it's a perfect 50-50 split across the body. Half the cells use the father's X, half use the mother's. But by pure statistical chance, some carriers end up with skewed inactivation. They accidentally silence the healthy X chromosome in a large majority of their muscle or heart cells.
Oh, no.
Yeah. When that happens, they become what we call manifesting carriers. They can develop real, sometimes severe muscle weakness or life-threatening cardiac issues themselves.
Which completely dismantles the old textbook teaching that female carriers are just silent vehicles for the gene.
It absolutely does.
So understanding all of this, the sheer size of the gene, the mechanical failure, the reading frame rule, it wasn't just an academic exercise. It became the exact blueprint for how researchers decided to fight back. And this brings us to the cutting edge of genetic medicine and honestly, easily the most fiercely debated therapies in medicine today.
Definitely. Let's start with 2016. The FDA granted accelerated approval to a drug called eteplirsen, marketed as Exondys 51. This is known as an exon-skipping drug, and the strategy relies directly on that reading frame rule we just discussed.
How so?
Well, if a patient has a mutation that disrupts the reading frame, this drug goes into the cell and chemically forces the splicing machinery to deliberately skip over another specific section of the gene exon 51.
Wait, so you are intentionally causing a second control deletion to fix the first one. You remove more genetic code to force that broken, severe, out-of-frame mutation back into a milder in-frame mutation.
Yes.
You are literally trying to medically turn severe Duchenne into milder Becker.
In theory, it is a masterpiece of rational drug design. But the actual approval of this drug was wildly contentious. The FDA approved it based purely on a surrogate biomarker.
And a surrogate biomarker means they didn't prove the drug made the patients walk further or feel stronger. Right?
Exactly. They only proved that muscle biopsies showed a minuscule increase in the production of dystrophin protein.
Right, because looking at the regulatory records, the FDA's own internal review team objected strenuously to approving it. They argued the amount of protein produced was so tiny it couldn't possibly provide a clinical benefit.
Yes, the internal dissent was huge.
Furthermore, this highly specific drug only applied to about 13% of Duchenne patients whose unique mutation could be theoretically corrected by skipping exon 51. The approval only went through because a senior FDA official directly overrode the staff-level scientific objections.
It really became a landmark case study in medical ethics and regulatory capture.
Yeah.
The core debate was, how much actual clinical benefit should an accelerated approval pathway require before a vastly expensive drug is offered to desperate patients?
We are seeing that tension play out in real time because patients and their families are naturally desperate for interventions right now, not 10 years from now. But we also face the incredibly harsh reality that genetic interventions carry severe, sometimes fatal risks. And we have to look at the tragic events of 2025 regarding a drug called Elevidys.
Right. Elevidys took a completely different approach. It is a gene therapy. It uses an engineered virus, specifically an adeno-associated virus, or AAV vector, to deliver a shortened synthetic microdystrophin gene directly into the patient's muscle cells.
So they basically hollow out a virus, remove its ability to cause disease, stuff the microdystrophin gene inside, and use the viral shell as a microscopic delivery truck.
That's exactly it. And Elevidys was granted accelerated approval in 2023. But by 2025, devastating reports emerged of several teenage boys with Duchenne dying of acute liver failure shortly after receiving the high-dose viral treatment.
And maintaining strict impartiality here as we look at the regulatory landscape, the Elevidys situation triggered a massive reckoning. It renewed intense scrutiny of high-dose AAV gene therapies across the entire pharmaceutical industry.
It did, because the immune system often recognizes these viral delivery trucks as foreign invaders and mounts a catastrophic attack, often targeting the liver where the virus accumulates.
It is a sobering reminder that successfully putting a gene into a cell is not the same thing as curing a disease safely.
Not at all.
We are witnessing a profound tension between the urgent life-or-death need to try something new and the reality that these delivery vectors can be fatal. This is changing the calculus for accelerated approvals across all of medicine.
Yeah, the safety policies being debated and drafted right now because of Elevidys will dictate how every single future gene therapy, whether it's for sickle cell, cystic fibrosis, or Huntington's, gets evaluated. Duchenne is the test case for the future of genetic medicine.
And while the scientific and regulatory communities wrestle with the FDA, there's an equally complex social debate running parallel to the science. For decades, the primary public face of muscular dystrophy in America was the Jerry Lewis Libre Day Telethon.
Right.
It ran for over 4 decades, finally ending in 2011, and it raised well over $2 billion.
And there is no denying the financial impact of that. I mean, that telethon funding is widely credited with paying for decades of the exact molecular science we've been unpacking today. The identification of the dystrophin gene, the early animal models, the patient care services. A massive portion of that was fueled by the money raised over those Labor Day weekends.
But the sources also detail how that same telethon was heavily condemned by disability rights activists. Like in 1981, a prominent activist named Evan Kemp Jr. wrote a widely cited op-ed arguing that the telethon relied entirely on pity-based fundraising.
Yes.
Critics argued that by portraying people with MD merely as helpless, tragic objects of pity just to extract donations from the public, the telethon did real, lasting harm to how capable, independent, disabled individuals were perceived and treated in daily society.
It sparks a really profound debate about the ethics of scientific funding. Do the ends, billions of dollars for vital life-saving research justify a means that disability advocates felt actively compromised their dignity and social standing?
Right.
It just shows us that the history of this disease isn't just written in laboratories with microscopes. It's written in regulatory hearings, courtrooms, and social movements.
Which brings into focus why all of this matters so deeply today. Duchenne alone affects roughly 1 in every 3,500 to 5,000 male births worldwide. That makes it one of the most common serious genetic disorders in humans.
And as patients live longer due to better respiratory care, the very nature of the disease is actually shifting. You see, the heart is a muscle too.
Oh, of course.
Yeah. So dilated cardiomyopathy, where the cardiac tissue is eventually replaced by that same scar tissue, is now the leading cause of death in DMD patients as they survive into their 30s. The seatbelt fails in the heart, just like it fails in the legs.
There is one final thought we need to leave you with, a choice we are facing right now as a society. We mentioned earlier that when the muscle cell membranes rupture, the contents of the cell leak out into the bloodstream. And one of those leaked proteins is an enzyme called creatine kinase, or CK.
Normally, CK levels in the blood are quite low. But in a child with Duchenne, a simple blood test can show CK levels 10 to 100 times the normal limit. It's often the very first clue that something is wrong, leading to a diagnosis long before a child shows any outward signs of clumsiness or weakness.
Because it is such a simple, cheap blood test, there is an ongoing, intense debate about newborn CK screening. We possess the technology right now to screen every single baby born for Duchenne muscular dystrophy on the very first day of life.
But the bioethical dilemma is immense. I mean, we've established today that the current genetic treatments carry severe, sometimes fatal risks. We've established that we do not yet have a definitive, completely safe cure.
Right.
We also know that a child with Duchenne will walk, play, and seem perfectly healthy for years before the mechanical strain finally breaks down their regenerative capacity.
So the ultimate question to chew on is this. Would you want to know? If it was your child, would you want to know on day one that they have a fatal genetic disease years before any symptoms appear and years before any truly safe treatment can actually help them?
It's a question that sits at the very center of where the entire field of genetic medicine is heading. The technology gives us the foresight, but we lack the tools to change the outcome safely.
We started this deep dive thinking about how movement builds us up, how adaptation makes us stronger. But the reality of our genetic code is that sometimes the molecular seatbelts we rely on to survive gravity simply aren't there. And while a standard physical exam might not show the defect early on, the microscopic landscape reveals a battle that is defining the future of how we will treat all human disease. Keep that in mind the next time you think about the simple, impossibly complex act of taking a step.